Welcome to the Applied Biosystems Taq Talk video series, where we break down real-time PCR to make it easier for you. Did you know that qPCR can be used in a variety of different applications and research areas? Let's talk about some of the most common ways that people use this awesome technology. Most notable on the list of applications is looking at relative expression levels of different gene targets. Researchers compare two or more samples, say cells from untreated and treated animals, to determine if and how much the expression of their gene of interest is changing. Using Applied Biosystems real-time PCR systems in combination with either custom or pre-designed assays, researchers can look at either messenger RNA or small RNA targets, including specific microRNAs. So, what else can real-time users do with their instrument? So many applications, so little time. A common one is using a standard curve to calculate absolute or relative copy number of a DNA target. This could be a pathogen in food or animal samples, a microbe in water, or any other detectable nucleic acid sequence. Speaking of food, some labs use qPCR to calculate % GMO in food samples in order to ensure that they meet legal standards. Copy number studies on genomic DNA targets are quite important to labs that typically need to either segregate animals with different copy numbers of a transgene or detect and quantify duplication or deletion events associated with a particular phenotype. Keep in mind, I've only mentioned some of the more common possibilities. The fact is, pretty much any application that requires quantifying nucleic acid targets can be done using real-time PCR. Well, what about experiments that aren't quantitative? The real-time system has you covered there as well. Perhaps most notable is the allelic discrimination assay, which detects specific single nucleotide polymorphisms in DNA samples, and then segregates those based on their homozygous or heterozygous genotypes. In a situation where a researcher wants to merely detect rather than quantify pathogens, as with SARS-CoV-2 testing, our presence-absence application and associated detection reagents offer a comprehensive solution for even low-expressing targets. There's also an option on many Applied Biosystems real-time PCR instruments to run high-resolution melting experiments, which can be used to check % methylation of short DNA target regions, or to discover small sequence differences among multiple samples. Besides nucleic acid-based analysis, real-time PCR is increasingly being used to look at proteins. Applied Biosystems Taq protein assays combine the protein binding capabilities of antibodies with the relative quantification capabilities of qPCR. We also offer protein thermal shift reagents and software for screening proteins to measure their thermal stability in the presence of different ligands, mutations, medications, or buffer conditions. And don't forget, regardless of the application, Thermo Fisher offers a full suite of reagents, assays, and analysis software to help you get the job done. And that, of course, includes millions of pre-designed Applied Biosystems Taq assays for numerous applications, including gene expression, SNP genotyping, microRNA, and many more. For even more information about real-time PCR applications and technologies, go to thermofisher.com/qPCR applications. Thanks for watching. Check out more Applied Biosystems Taq Talk videos on our website at thermofisher.com/taqtalk.